We have studied about the PCR reaction, polymerase chain reaction. We learned about the reverse transcription polymerase chain reaction, that is RT-PCR. Now we are going to study about the types of PCR. So what are the types of PCR we have in the modern day technology other than the existing RT-PCR or the normal PCR? So why do we need this different types of PCR? So what is the use and application of it from the rest of the PCR? So that is what we are going to learn. So first one is what we're gonna learn is conventional PCR. Conventional PCR is what exactly we learned, the normal PCR. So basically here we are going to take a sample from the any places like crime scenes or extracted DNA from any of the humans, hair strands or the bacteria, and this DNA is denatured with a temperature of 95 degrees Celsius, and then we are making it to anneal it with the primers, and then we are using Taq polymerase for the extension. So this is what exactly normal PCR we are using, and this is also called conventional PCR. Conventional PCR is used in the majority of the laboratories for the day-to-day PCR reactions to carry out, like we have it in the colleges, research institutes. Majority of the small scale laboratories use conventional PCR for the amplification of DNA, and even the forensic science also use the conventional PCR for the amplification of the DNA. So the conventional DNA is having this thermostable Taq polymerase, which the ingredient we are using here is a template of the DNA from the place of the sample where we are extracting the sample, and then we have Taq polymerase followed by the buffers and nucleotides and primers. So this is what we are going to use in conventional PCR. So after this we are going to learn about the second type of the PCR, that is reverse transcription PCR, or also called reverse transcriptase PCR, also called RT-PCR. So RT-PCR is very exactly the difference in RT-PCR only because we use the sample of RNA. That means who has RNA right now? The viruses. So usually whatever we extract from the viruses, which has the RNA as a genetic material, we extract it and then we use it as a single strand, and that we will produce a complementary DNA with the reverse transcriptase enzyme. And once we are producing a reverse transcriptase complementary DNA strand, we will having a RNA and DNA hybrid. Once this RNA DNA hybrid is formation, we need only DNA, right? So we are going to destroy the RNA with the RNase H. Once we destroy the RNase H, we will have the single stranded DNA, and this single stranded DNA will go for the PCR reaction. So what we spoke in the previous type, conventional PCR, this single stranded RNA will go for the conventional PCR, and we will make the multiple copies by the amplification method. So at the end of the reaction of the reverse transcription, we are going to convert the RNA to the DNA, and we will make the multiple copies, and these copies can be used for the assays like ELISA, enzyme linked immunosorbent assay. Like if that DNA is present, since we know this DNA is belonging to the viruses, it gives the positive, and if it is, and it gives also the understanding about the concentration. So what concentration it is present, higher concentration, that means the higher amount of the virus's infection is there. If it is lower quantity is there, you will have the lower concentration. So concentration also will be able to understand with the further RT-PCR studies. So once these are the majority types of the PCR used, the conventional and the RT-PCR, these are the basic PCR technique is used in the majority of the times. But now we have something called real-time PCR, also called quantitative PCR. So usually people get confused with the reverse transcription PCR and real-time PCR, because it happens that people call both the PCR as the RT-PCR, but then the reverse transcription PCR and real-time PCR are very different components, because reverse transcription PCR we are using RNA, where exactly real-time PCR we are using DNA, but it has an advanced technique which makes the PCR more advanced or the modern technologically used. So how exactly it goes in the real-time PCR? So in the real-time PCR, so in the majority of a PCR, when the reaction is happening, let's say in the beginning and the end, there is no understanding between the pathways. So whatever denaturation, annealing, extension, all these processes are not understood. We do not know what is happening in the PCR machine or the PCR instrument. We just carry out the reaction. We expect it to be happening, and at the end of the reaction, we expect the result to become. But what happens is, in the PCR reaction, we use a microliters. It is not even a drop. When we use it in the pipettes, it is not even a drop. So that is why we expect our ingredients to be there, like buffers and the magnesium chloride and then the enzymes. All of them we put in the 0.1 microliters, 0.2 microliters, and we use the Eppendorf tubes for it. And throughout the reaction, we do not know what has happened in the Eppendorf tubes. Once the reaction is done, we run the gel electrophoresis. At that point, we get to know our DNA is not amplified. We get to know our DNA is not amplified in a right manner. So in that process, it happens that a person runs the PCR and then runs the gel electrophoresis. It takes around 2 days to do it, and we do not know where exactly we went wrong. So each time we have to tackle the problem with the solution we used, concentration we used, all those things. So to reduce those kind of a problems, we took a real-time PCR. So what exactly real-time PCR does is it shows the how exactly the process happens. So here we have SYBR or cyber PCR we say, and the Tachman PCR. In the cyber PCR, basically you see here we used a green kind of a fluorescence over here. What happens is, once the denaturation happens of our DNA, at this time our fluorochromes will not be binding to it. Once the second is our primer binding, that is the annealing process. At that point, there are chances of the fluorochromes binding, but here the binding happens to the hydrogen bonds between the two nucleotides. So when the extension happens, now let's say during the extension, complementary DNA is readily added to the other strand, the template strand, right? The complementarily added, the nucleotides. So here during the extension, each hydrogen bond will be bound with this fluorochrome till the end of the reaction, because extension is nothing but the hydrogen bonding happen till the end of the reaction. So throughout that hydrogen bonding addition, we will be having this fluorochrome giving the color. So this process, during the process we can see this color formation. With this we will understand, okay, now the denaturation process is happening, because we are not seeing the fluorochrome, because in the denaturation we are breaking the bonds between the templates. Now the RNA, the little fluorescence is seen when the annealing happens, because primers have only 3 to 5 to 10 hydrogen bonding, but the extension of the DNA happens in the complementary bonding. Here we will have the SYBR green fluorochromes, we also call it as a cyber fluorochromes, readily binding to the hydrogen bonding. So at the end of this reaction, from the beginning to the end, we know what process a PCR reaction is going through, which is not possible in the conventional method. And this is the what exactly we have upper hand. So if we are not if you have gone through any of the processes wrong, let's say extension is not happening, the annealing is not happening, we know at that point of time, so this reaction is not happening, so there should be some issues we have to tackle in this reaction, and we have to see what buffers could be used at this time. So all these things could be tackled or directly tackled in this kind of a PCR reaction. In the real time, we have one of the fluorochrome used cyber, so as if we have gone just now. The one more is the Tachman. In the Tachman, in this case, there is one special case, like you should know your DNA well in advance, and that DNA when you use, you are taking another sequence of DNA which has a fluorochrome and quencher. So what happens, this probe is having specific sequences, you know this limitation over here, you should know your DNA sequences well in advance. So this probe will directly bind to the your DNA complementary strand. So what happens when the fluorochrome is near the quencher does not allow the fluorochrome to fluorescence. The quencher's work is not allowing the fluorochrome to fluorescence. So what happens, once the extension starts, the fluorochrome will be detached, and it shows the fluorescence, and that time we know that the PCR reaction has happened and it has been completed. So in the beginning, without doing any of electrophoresis, we know at the end of a reaction that this reaction has happened successfully, and this is the where we use real-time PCR. So real-time PCR, also called as quantitative PCR, because we know the reaction is happening. The signal emission, the fluorescence emission, if you are able to detect, that gives the concentration of the DNA also. If the DNA concentration increases in the subsequent cycles, we know that concentration, fluorescence color emission is directly proportional to the concentration of the DNA. So this is what exactly we get to know about the real-time PCR. Now the last PCR technique we are using is colony PCR. So this is a very modern PCR technique. So what exactly this does is, so every time whenever I have to do PCR, the basic problem I have is I have to extract the DNA. So I have to use my blood. If I want to extract my DNA, I have to use my blood and I have to do the centrifugation. I have to extract my DNA, I have to purify my DNA, and then I have to run the PCR reaction. But now let's say when we use bacteria, there is a problem. Sometimes the extraction process becomes so tedious. So to avoid that, now instead of extraction, we are directly using the bacterial colony and we are inserting into the Eppendorf tubes with the PCR reaction. Here this PCR, colony PCR, able to directly amplify the DNA in the bacterial colony without a human manually application of the extraction of DNA. So we can just directly use the sterilized toothpick or any of the sterilized instruments for the removal of the colony from the Petri plates. That colony we can insert into the Eppendorf tube, and once we have inserted the Eppendorf tube, we can add the PCR reaction mixture, and after that we can run the PCR. So colony PCR does not need the extraction of DNA. So these are the techniques we use. These are the types of the PCR we use in the modern day techniques.